Good morning, students. Our today's topic is pedigree analysis. An analysis of traits in several of generations of a family is called the pedigree analysis. In this analysis, the inheritance of a particular trait is represented in the family tree over generations. In simple words, this is the chart of the family history showing the inheritance of a particular character. Again, this is the chart of a family history showing the inheritance of a particular trait. It is known as pedigree chart. Some of the important standard symbols used in the pedigree analysis have been shown in figure 5.13. So I will explain these terms. So first, the square male is represented by a square, female is represented by a circle. The diamond, this shows that sex is unspecified, whether it is male or female, not clear. If it is colored red, that means that individual is affected individual. The line between the square and the circle shows mating, that is the meaning marriage. This is known as horizontal bar between the square and the circle, which represents mating. If two lines are there, then this shows the mating between relatives. Relatives means very close persons, very close relations. This type of mating is known as consanguineous mating. Learn this term, consanguineous mating, that is a mating between the close relations. Next, square circle, that is the marriage. Afterwards, that is shown the F1 generation, the offsprings. From the left side, the first child is a girl and second one is the son. So parents above and children below in order from birth left to right. Next, the red box is shown in red color, which shows that the child is son and affected with disease. The next topic is Mendelian disorders. Genetic disorders may be grouped into two categories, that is, first is Mendelian disorders and chromosomal disorders. Mendelian disorders are mainly determined by change in a single gene, that is, when mutation occurs in a single gene, that is a Mendelian disorder. The pattern of inheritance of such Mendelian disorders can be traced in a family by a pedigree analysis, that we have already discussed under pedigree analysis. So Mendelian disorders are hemophilia, in which the clotting of blood is affected, cystic fibrosis. In this, the mucus becomes thick and sticky and block the tubules of respiratory tract and sometimes the digestive tract also. Next, sickle cell anemia, in which the RBC change their shape from biconcave to sickle shape. Color blindness, in this the person is unable to distinguish between the different colors. Phenylketonuria, this we will study. So, by pedigree analysis, one can easily understand whether the trait in question is dominant or recessive, or whether it is sex-linked or autosomal. So here figure 5.14, A part, in this the circle is red and square is normal. That means female is affected and male is normal. The female is transferring that disease to the male also and to the female also, to the male also and to the female also. So this clearly shows that this is not the sex linked. Sex linked means when it occurs only in one sex only, either to the male only or to the only females, mainly to the males. So because the disease occurs to both the male as well as to the female, so this is autosomal trait or autosomal affecting trait. So From this cross, it is clear that it is an autosomal trait. And it is dominant trait. Why it is dominant? Because it is present in the mother and then it comes into the progeny. So it is a autosomal dominant trait and autosomal dominant trait. Next, B part. Here the parents are normal. Parents are normal, but their offspring show the disease. This shows that this is a recessive trait, but it is autosomal. Why? Because this trait occurs in both the male as well as in the female offspring. So B chart shows that it is autosomal recessive trait, whereas A shows that it is autosomal dominant trait. So now the next disease is hemophilia. This is a Mendelian disorder. In male, actually the normal genotype of a male is XY. XY means normal male. When X is hemophilic, X has the H allele, H represents the hemophilia, then this is the affected male. So this is the genotype of the male affected with hemophilia. For the normal female, genotype is XX. If only one allele is present on X, that is a carrier. And if both the X contain the H, that is the affected hemophilic individual. Now this is a cross. When the female will be affected, the female will be affected when at least the mother will be carrier and father will be hemophilic. If father is hemophilic, mother is carrier, then only the chances is that the female is, or the F1 offspring is affected female. Actually, it is not possible. Chances are very less, because for that the male is not viable. That male cannot survive up to the maturity period. So in this, the chances of becoming a female affected with hemophilia are very rare or very less. And next is the male becoming affected. Then the genotype will be XHY. This X can come from the carrier female. Here female is the carrier and the male is normal. Normal male with the carrier female produces the affected male progeny. So male is affected from the carrier mother, carrier female. This is possible. Why? Because the carrier female can transmit the gene to the son. Hemophilia is a sex-linked recessive disease which shows its transmission from unaffected carrier female to some of the male progeny. This means from the carrier female it goes to the male progeny. In this, a single protein, that is the part of the cascade of protein involved in the clotting of blood, is affected. Means one protein which is required for the clotting of blood is not present here, so blood clotting does not occur, and in this individual continuous non-stop bleeding occurs. The heterozygous female for hemophilia transmit this disease to the sons. The possibility of a female becoming hemophilic is extremely rare, because mother of such a female has to be at least carrier and father should be hemophilic, which is not possible, which is unviable in the later stage of life, as I have already explained to you. The Queen Victoria was the carrier, so she transmit that gene to her son and shows a number of hemophilic descendants. She passed that gene to many of the male progeny. It is also known as royal disease because it was detected earlier in Queen Victoria family. Next is sickle cell anemia. It is represented by two alleles, HBS and HBA. If both are HBS, that is the affected individual. If HBS and HBA, that is carrier. S shows the sickle cell anemia, that person is affected. HBA means normal individual. It is an autosomal recessive trait. This trait is transmitted from parents to offspring when both the partners are at least carrier for the gene. Because it is recessive, so at least both the parents should be carrier for this disease. Next, this is mainly caused by the substitution of glutamic acid by the valine at the 6th position of beta globulin chain of hemoglobin molecule. Here diagram is shown which shows the beta chain of hemoglobin. At the 6th position, the glutamic acid is changed with valine. The glutamic acid is one amino acid that is shown by GAG, which has the code GAG, and valine has GUG. In this, A is changed with U. The substitution of amino acid in the globulin protein results due to single base substitution at the 6th code of beta globulin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization, undergoes change because it has now less oxygen carrying capacity, which changes the shape of RBC from biconcave disc to sickle-like structure. Phenylketonuria. This disorder is present from birth itself. It is an autosomal recessive trait. The affected individual in this lacks an enzyme which converts the phenylalanine, that is an amino acid, into tyrosine. Means in this one enzyme is missing which usually converts phenylalanine into tyrosine. Because of this, the phenylalanine accumulated in their body and finally converted into phenylpyruvic acid. Because of the accumulation of this phenylpyruvic acid in the body, it causes mental retardation. These are also excreted through urine because of their poor absorption by the kidney. Now, the chromosomal disorders. These disorders are caused due to absence or excess of one or more chromosomes. So, during cell division, the chromatids fail to segregate, which results in loss or gain of chromosomes. This is called aneuploidy. This condition is known as aneuploidy. Example is Down syndrome, in which one extra chromosome occurs in chromosome number 21. Actually, pair of chromosome is there, but here 3 chromosomes are present. Turner syndrome, where loss of one X chromosome occurs. So, this occurs because the chromatids fail to segregate at the time of cell division. Sometimes the whole set of chromosome in an organism increases. So, this is known as polyploidy. And in aneuploidy, 1 or 2 chromosomes are added or deleted. But in polyploidy, the whole set of chromosome is increased. This often occurs in the case of plants. In humans, total 46 chromosomes are present. That is 23 pairs of chromosomes are present. Out of these, 22 are autosomes and 1 is the sex chromosome. So sometimes when one extra chromosome is added, that condition is known as trisomy, and when one is deleted, that is known as monosomy. So next is Down syndrome. It is caused due to the trisomy of 21st chromosome. That means here 3 chromosomes are present at the 21st position. This disorder was first discovered by Langdon Down. On his name, this disease name is given Down syndrome. The affected individual in this case has the short stature, round face, furrowed tongue, and partially open mouth. And they are mentally, physically, and psychomotorly retarded persons. Next is Klinefelter syndrome. This genetic disorder is caused by the extra copy of X chromosome. So, in these individuals, they have XXY genotype. That is, their total number of chromosome is 47. They are phenotypically male. Phenotypically means from the outer appearance, male-like structure is present. Such individuals has masculine development. Their body is very muscular, and sometimes, and they have feminine developments also, that is the development of breast. This condition is known as gynecomastia. Learn this word. Such individuals are sterile. Sterile means which cannot reproduce. Next is Turner syndrome. Such a disorder is caused by the absence of one X chromosome. So individual have 45 chromosomes and sex chromosome XO, that is one chromosome is missing in this. Such females are sterile. Sterile means they cannot reproduce. The ovaries are rudimentary. Rudimentary means only small structures are present, they are not functional. And other features like lack of secondary sexual characters is also present. They are phenotypically female, that is, from the outer appearance, female-like structure is present.